So my name is Fokhrul Hossain. I am in the Department of Genetics, LSU Health Sciences Center. So the good thing about presenting later on is that most of the introduction has already been given. So I'm not gonna spend more time on the introduction part. I'm gonna skip some of the part. Mainly Dr. Shu mentioned many of the stuff already I was planning to say. So as you all know that the breast cancer is the most common cancer in the woman. And this is the 5th cause of cancer death worldwide. Okay. And here you can see that the incident rate of the breast cancer, is widely, in incidence rate is widely across the globe. However, if you look at the mortality rate, you can see the certain part of the globe has a higher mortality rate than the others. So this suggests you the lack of access to the healthcare as well as the breast cancer disparities. So WHO or the World Health Organization mentioned in 2020 there have been 2.3 million women have been diagnosed with breast cancer and with 685,000 deaths globally. So in the US also, the breast cancer is the most common cancer in women, and this is the second cause of cancer mortality. Already Dr. Sam mentioned these things. So I'm going to give you very briefly the different subtypes of breast cancer. Although already we have learned a lot about the triple negative breast cancer, but the other form are the luminal A, which is are the estrogen receptor positive, progesterone receptor positive, and then HER2 negative. That accounts for around 60 to 70%. And the luminal B, which are ER, PR, and HER2 all positive, those are accounts for around 5 to 10%. And then the HER2 enriched, these are around 10%. And the triple negative, you already heard a lot about the triple negative and subclassification. Those are accounts for around 15 to 20%. So as all the other speakers are saying that obesity is a complex condition and it's involving the accumulation of fat in the body. And also Dr. Shu and Dr. Mazumdar already mentioned that obesity is linked with the chronic inflammation. And that's why it's linked to the many diseases, including cancer. And here is the list for the NCI. There have been 13 cancers have been associated with the obesity, and breast cancer is one of them. And so there are many factors that are involved in the breast cancer progression, such as the high estrogen level in the blood, higher insulin level in the blood as well, the body shape, body composition, weight loss, and weight gain. So I was planning to discuss in details about this epidemiology study, but Dr. Shu already mentioned that how the menopause and the BMI are correlated. I'm going to give you the very brief overview on this one. So, the most of the studies report that the obesity associated with the lower estrogen receptor ER positive cancer risks before menopause. On the contrary, obesity is associated with higher risks of premenopausal ER negative breast cancer as well as the triple negative. Here you can see the forest plot as well. Okay. And next, here you can see what happened in the postmenopausal cases. And most of the studies suggest that the obesity associated most strongly with the postmenopausal hormone receptor positive breast cancer risk. And on the contrary, the risk of ER negative breast cancer and the TNBC is minimally or inversely associated with obesity after menopause. So there have been lots of study there as well. And also there is lots of epidemiology study suggesting that obesity is associated with higher breast cancer mortality as well, as Dr. Shu already mentioned during his presentation. So, now I'm going to talk about a little bit about the mechanism and how the obesity is related, induce the cancer progression. So during obesity, as you know, there's one of the hallmark is the hypoxic condition, which activate the HIF-1 alpha and it decrease the adiponectin. On the contrary, it's increase the leptin. HIF-1 alpha also increase the angiogenesis by promoting the VEGF. So this alter the leptin and adiponectin ratio, promotes the pro-inflammatory immune cells infiltration and release of the cytokines. And in addition to that, the dying adipocyte release the free fatty acid, which can bind to the TLR4 on macrophages, myeloid cells, or adipocytes to activate the NF-kappa B pathway, and which in turn upregulate the secretion of inflammatory cytokines. So the NF-kappa pathways also can increase the aromatic in expression. And as you all know, the aromatic expression which can convert the androgen to the estrogen in the body. So therefore, in the obese condition, there is lots of circulating estrogen in the blood. So next slide, I'm going to talk about a little bit about the, how obesity changes the tumor immune microenvironment. And here in the cartoon, you can see, this is the obesity in the lean woman, where you can see a expression of the anti-inflammatory factors and anti-engineering factors as well. But however, in the obese woman, this has been changed. So what happened is during the obesity, the increased pro-inflammatory as well as the pro-angiogenic cytokine increase the, and you can see here, increase the angiogenesis. It can increase the stem cell expansion. It can increase the metastasis. It can increase the invasion as well. So one of the issue in the obese condition is that the mediator of anti-tumor immunity, such as the CD8 T cells, NK cells, or dendritic cells, are decreased in the obese condition. On the contrary, the immunosuppressive cells, such as MDSCs, which are the myeloid-derived suppressor cells, or tumor-associated macrophages, are increased within the tumor microenvironment. As by their name, so this immunosuppressive environment help the tumor cells to grow within the tumor microenvironment. So another thing is obesity also increase the insulin-like growth factor 1, and which in turn increase the tumor proliferation with many pathways, including AKT1 and other pathways as well. So overall, you can see that during obesity, there is a change in the tumor microenvironment, and which help the tumor to grow. So now I'm going to share some of our published and unpublished data with you. So in collaboration, we reported that the leptin produced by the obese altered adipose stem cell promotes metastasis, but not tumor genesis. So we use two different models. One is the orthotopic model, and another one is the PDX model. And in the both model, you can see that the tumor growth remained the same. However, then when you look for the metastasis, the area of metastasis in the lung, you can see that the mice injected with the obese adipose stem cell has a higher number of metastatic cell in their lung. Next, we also measure the circulating tumor stem cell in the blood, and you can see this the mice injected with the obese adipose stem cell has a higher circulating stem cell as well as the HLA marker in the blood. So next, we also did the immunohistochemistry from those mice, and you can see there is a upregulation of the CD4 expression in the obese group, and then we do the immunohistochemistry with a confocal microscopy to find out that this CD44 cells also express the fatty acid transporter. And here you can see there is although we don't have the quantification here, but there is a more expression of CD36 in the obese mice tumor compared to the lean one. And next, we also look for the CPT1 expression. As you all know, that is the rate-limiting enzyme of the fatty acid oxidation pathway. And you can see here, similar to the CD36 expression, CD4 cells also express the CPT1, suggesting that the fat-related metabolism are increased in this tumor. And this is another example. We have done separate from the other experiment, where we can find that. Let me change this one. So you can find that in the obese tumor. So here we use two different markers. Another marker for the cancer stem cell. It has been also reported that the CD90 can also be a cancer stem cell marker. And you can see when you stain for the CD90 and CD36, you can see the co-localization higher in the obese condition, suggesting that these cancer stem cells are also enriched in the fatty acid uptake as well. Okay. And so here is another, then we ask the question, so what are the, they ask the question, what is the role of obesity in the tumor TNBC progression? So we used a syngeneic mouse model. So we use the 0321 cells as Dr. Sam mentioned a couple of minutes ago. So we fed our ABP female mice for the Western diet and as well as the control diet. And we could see by their, picture, there's obese, obviously the Western diet induce obesity. And here is the weight. And then also we weighed the liver and the visceral fat, which are significantly increased in the Western fat diet. Then we isolate the liver, blood, and visceral fat to check for the immune cells in those mice. So we see there are some changes in the immune cells. Mainly we see the changes in the MDSCs and macrophages. However, we didn't see any significant differences in immune cells just after the obesity growth. So after that, we inject the syngeneic mouse cell, the 0321 cell, into the memory fat pad of the lean and obese mice, and follow the tumor growth for another 3 weeks. And here you can see that these obese mice are blue. That has an increase in the tumor volume. And then after that, we digest the tumor, get in the single cell suspension, and did the flow cytometry analysis for the immune cells. And you can see there is a trend of decrease of CD8, CD4. However, due to the lower number of N, we didn't see the significance in our experiment. We didn't see much changes in the dendritic cells. However, we did see there is upregulation of MDSCs and macrophages. As I mentioned earlier, these are the cells are immunosuppressive cells, which can suppress the immune cells so that the tumor can grow happily. Okay. And next, we isolate the tumor-associated tumor MDSCs from the lean and obese mice. So, and then we measure the metabolic profile using the seahorse analyzer. And these are the OCR, the oxidative consumption rate, as a measurement of the oxidative metabolism. And this is the extracellular acidification rate as a measurement of the glycolysis. And see that the glycolysis, we didn't find any significant changes here. But here, in the OCR, the oxidative metabolism, you can see that the obese tumor MDSCs had the higher basal OCR as well as the maximum capacity. And of course, they have the higher reserve capacity compared to the lean MDSCs. So finally, we asked the question, as we all know that the tumor MDSC's main job is to suppress the T cell proliferation. So we did the T cell proliferation assay to see whether these MDSCs can suppress the tumor growth or not. So in our model, just to mention here, that is, our MDSCs is not that much of suppressive compared to the other model. And you can see that here, let me move these things a little bit. So lean MDSCs, as I mentioned, they're not very suppressive compared to the other model. But when you use the MDSCs from the obese mice, there is a significant decrease in the T cell proliferation. So overall, so next we also did the RNA-seq from the lean and obese mice. And this experiment, the analysis is ongoing. And here you can see that the obesity-relevant pathways are upregulated in the obese tumor. And we have the upregulated genes and the downregulated genes as well. So we are doing a lot of other analysis right now in collaboration with our bioinformatician. And hopefully we'll be able to publish pretty soon. So the overall, in our model, using the syngeneic mouse model, we can say that the Western diet, as expected, can induce obesity, and it can enhance the syngeneic TNBC tumor growth. And obesity increases the tumor-infiltrating myeloid cells, mainly MDSCs and macrophages. And this suggests that the accumulation of the immunosuppressive environment, as I mentioned earlier, and this helps the tumor to grow. And the obesity increases the oxidative metabolism and augments the immunosuppressive function of MDSCs. So I have not mentioned here, but in another parallel project, we are doing the characterization of lean and obese TNBC patient from the state of Louisiana. So hopefully we'll report in near future. So with this, I'd like to thank our collaborator and funding for supporting our research. Thank you all.
Thank you so much for this great presentation. Are there any questions?
So Fokhrul, what is the difference in the MDSC and macrophages, the change in the lean versus obese versus after tumor engraftment in both the cases? Is the change is drastic? Is it increased, the MDSC and macrophages, compared to the before engraftment or after engraftment?
Yeah, that's a very good question. So remember, that's why we did our analysis before the tumor in engraftment, and at that time we didn't see much changes. We also looked for the MDSC has two subsets. MDSC has two subpopulation, the DMDSC and MDSC.
Yeah.
So we didn't found much difference before the tumor injection, but after injection, there has been a lot of changes. So, I mean, as you know, the MDSCs are immature myeloid cells. So with the influence of the tumor cytokines and chemokine, this becomes suppressive. So that's the reason why you see the significant changes after the tumor injection compared to the without tumor.
Thank you.
Excellent.
Well, I have a question related to the leptin.
Sure.
Is the leptin receptor highly expressed in breast cancer? And if yes, then whether the
Yes.
expression associated with cancer prognosis?
Yes, that's true. So in that experiment, we collaborate with Dr. Bruce Bunnell. I mean, in the paper, they have done a lot of other experiment as well. They knock down the leptin, and we see that, tumor growth has been changed and all these things. But our focus is to whether, it can. So, but the overall finding of our paper is that it mainly induces the metastasis.